In today's video, we're going to start Chapter 3, and Chapter 3 is the first of the chapters about classical conditioning. You might have noticed by either looking at the syllabus with our course outline or looking at the textbook itself, there are actually two chapters on classical conditioning. The first is all about foundations, and the second focuses in on specific mechanisms. So we're starting with our foundations, and so a lot of what we're going to talk about today might end up being fairly familiar to stuff that you've done in the past regarding classical conditioning. We'll start deviating from stuff that you will have seen in other classes a little bit more as we get into things like mechanisms. For our outline for today, we're going to start with a little bit of history on classical conditioning, where it came from, the usual stuff. Then we'll move on to some experimental situations. That's pretty much what we're going to cover in this lecture. Next lecture, we'll get into things like excitatory and inhibitory Pavlovian conditioning methods, as well as the prevalence of classical conditioning and where we see it nowadays. Now, up to this point, we've talked about habituation and sensitization, which involves just learning about one stimulus. Now, if we just had one stimulus and that was the only way we could learn, we'd be fairly restricted in what we could learn. The world is a lot more complicated than a single stimulus could ever hope to encompass. In the real world, certain events are going to occur together or co-occur. And so we're going to be looking at classical conditioning as event-event learning, where we learn about two events that tend to happen together. This doesn't mean that classical conditioning is the most complex or the be-all and end-all of all possible learning options, but it is at least another step towards complexity beyond what we dealt with already with habituation and sensitization. So we're going from one stimulus to two stimuli that are associated in some way in the real world, or in the lab, as the case may be. Now, this learning can be used to help us predict events in our environment, to help us more effectively interact with our environment. Now, these events can either co-occur, sort of happening together at the same time, or they can happen one after another, maybe with a cause and effect type of relationship. But whatever the case, the association or the linking of these events is something that we can learn about. As an example of things that need to occur in a specific order, you can't walk through a door until you've opened it. If we're looking at things that tend to co-occur, maybe you can use cues of somebody in the first week of classes who's staring at their phone or a map, and they approach you. You might understand that they're looking for directions. All of those things co-occurring tell you one thing, that they're probably lost and don't know their way around, which is somewhat less of a problem nowadays, I suppose. So this brings us to classical conditioning. And classical conditioning is the simplest mechanism for learning about relations between two distinct events. Classical conditioning is useful because it allows animals to predict or anticipate events. So maybe you know where to find food or shelter in a specific area. Maybe you know that pine trees offer good protection in the snow, so you're going to look for a pine tree or something like that. Classical conditioning can be involved in making a response, so it can activate a behavior. If we have some kind of excitatory association where we do something in response to a particular stimulus, we would be making a response. But we can also see not making a response. So classical conditioning can be inhibitory. So if you have this urge to not do a response to a particular stimulus, like, say, looking for food if a predator is present. So you cannot do a behavior that you would usually do otherwise when a particular stimulus is present. There are lots and lots of uses for classical conditioning, so we're going to touch on a couple of those today as we start working through this chapter. Now, as you've come to expect, we can't start talking about classical conditioning without talking about the history of classical conditioning. And that, of course, brings us to Pavlov. something interesting that I learned through teaching this course is that Pavlov wasn't the only person who was looking at what we now call classical conditioning at the time. There was another researcher called Whitmire who was looking at knee-jerk reflexes in college students. So if they had a particular sound and then they would hit the tendon on your knee that causes you to sort of kick out your leg, they could then train or condition a classical conditioned response where you wouldn't have to keep hitting the tendon. You could then just play the sound that they had come to associate with that knee-jerk response, and they would jerk their leg. The interesting part here is that even though Whitmire was doing this research at about the exact same time, they didn't end up investigating any deeper into what was going on. So we never actually talk about their methods. If there's anything we can say about Pavlov, it was he took an idea and ended up running with it and marketed it fairly well. So what is this idea that Pavlov had? So originally Pavlov was a physiologist and was specifically studying digestion. Their lab was using methods to collect the digestive solutions of dogs to sell to basically make extra money for the lab. So they had found a way to harvest salivary and digestive secretions from dogs, and those secretions were desirable for other markets, and they could do it in such a way that the dogs could continue living normal lives for long periods of time. So they were set up to do that kind of as an extra way to make money. The classical conditioning side of things was actually an accident, where they found that dogs would start to salivate or produce larger quantities of digestive juices, even to just the sight of food. It didn't happen just while they were eating, it would happen in advance. It was as though they were expecting or predicting the arrival of food. So they called these anticipatory stomach secretions, or sometimes called them psychic secretions because of that predictive nature. And the general public really didn't care about these psychic secretions. They weren't interested. The research, when it was presented that way, didn't really get anyone excited for it. And so this is where Pavlov did something a little bit different that actually made this important. He made the connection and figured that we could actually use these anticipatory secretions as a method to study associative learning, and specifically the mechanisms of associative learning that might be within our nervous system. Now that was a question that people were interested in. So they could, use something that nobody cared about, but use it in a way to study something that people did care about, and it made them care indirectly about Pavlov and his salivating dogs. So his change in strategy, or maybe even his marketing, is what ended up making him the name that we see associated with classical conditioning, and led to classical conditioning being so important to the study of learning today. Now, in Pavlov's lab, he had two major researchers, two students working under him who studied these salivary responses in slightly different ways. And both of them are really important because they lead to how we understand classical conditioning today. Now, we'll have to pardon my pronunciation of Russian, but the first of these students was Wolfson, and they were interested in studying salivary responses. And to do this, they would place different substances in the dog's mouth to see if they could elicit some kind of salivatory response. So they used things like dry food, wet food, sour water, or even sand to see how much the dog would salivate in response to those substances. Then, if they did that enough, if there were enough presentations where the dog would experience those different substances and what they tasted like, what they felt like in their mouths, eventually just the sight of any of those substances would be sufficient to elicit salivation. They learned to associate just the sight of those objects with the taste and the texture of them in their mouths. Now, Wolfson focused on actual substances, whereas Snageyski, Snaisky, yeah, that sounds about right, focused on artificial substances. So instead of just giving the dog wet or dry food, they would give them sort of artificially engineered substances, things like sour black water. So they would have water that was, say, made sour with lemon juice, and then they would artificially change the color so that it's a novel thing that they would never have really experienced before. And then they did basically the same thing, give the dogs that sour black water, and eventually the dogs would start to salivate just to the sight of any black liquid. Now, for all of these different substances, the sour black water or the dry food, the wet food, whatever they were using, these were all what we could call orosensory stimuli, meaning that they have a distinctive texture and taste. So when the dog had it in their mouth, there was something distinctive about those items, those substances. And in both cases, over time, the visual features of those orosensory stimuli would also come to elicit salivation, even without having those items in their mouths. So we're associating orosensory features with visual features to elicit the same response. Now, this whole setup is what we can call object learning. So this is where we learn associations between different stimulus elements of the same object. So with the food, you can associate the taste of the food with the sight of the food, with the smell of the food. Object learning means you're learning about all of the different elements, all of the different stimuli that make up that particular object. Now, this can actually be fairly problematic. If we're looking at, for example, here, say our sour black water or even our wet or dry food, we have this problem where you can't disengage or differentiate between visual stimuli and smell stimuli and taste stimuli and texture stimuli because they're all coming from the same object. Because of this object learning, everything is all about the same item, and we can't really get a good study of the mechanisms because we can't really pick apart which property is driving that association. If we can't separate them, we can't independently manipulate our stimuli. So you can't dissociate the texture from the taste in a piece of food. And this inability to dissociate those different properties is what led Pavlov to use different stimuli from different objects in the training. That's where we get the idea of using a tone or a bell or some kind of light cue as the other stimulus, as opposed to just relying on the visual appearance of the thing that you would then eat later. So Pavlov used a separate stimulus from a different source that could then be manipulated to better understand the mechanisms that were going on. So we use a new stimulus to get around this object learning limitation so that we can actually manipulate all of the different factors involved. For the next couple of slides, we're going to run through the classical conditioning paradigm, the standard way of looking at classical conditioning. Now, this should absolutely be reviewed for you by now, at least twice over from different classes, so we won't spend too much time here. But we have our before conditioning status, so we can look at terms and methodology before the conditioning begins, before we start pairing stimuli. We have our unconditioned stimulus, the US, and that's going to be some kind of environmental trigger or stimulus. In this case, some kind of food, so you get the dog meat. And then we have an unconditioned response, or UR. And the unconditioned response occurs in response to that unconditioned stimulus. So this would be how the dog reacts to food, in this case, salivation. And the important part to note here is there's no need for prior training with this unconditioned response because it is naturally going to come from experiencing that unconditioned stimulus. That's why they're both unconditioned. They don't have to be taught. They exist already. Now, for terminology, sometimes you'll see a slightly different version of this where instead of US, you'll see UCS. I know that the intro textbook was bad for that. US makes more sense in my mind, so that's what I use here. But if you're used to seeing UCS, it means the exact same thing. It's just sort of a preference. Our next important player before conditioning happens is our neutral stimulus, or NS. And this is some kind of stimulus that initially elicits no response. So it is something that doesn't cause a particular behavior. So a tone or a light doesn't make a dog salivate because it's a neutral stimulus. So here they have a metronome, you could use a tuning fork, you can use a visual cue, whatever it is, it can be a neutral stimulus. This neutral stimulus will become our conditioned stimulus, the CS, which is a stimulus that's then going to precede or signal an upcoming unconditional stimulus. So we will eventually learn something about this neutral stimulus by associating it with our unconditioned stimulus, and then it will become a conditioned stimulus. So before conditioning happens, it's a neutral stimulus. After conditioning has happened, we're going to call it a conditioned stimulus. And we're usually going to transition from calling it a neutral stimulus to a conditioned stimulus once that conditioning starts, which we can see on the next page where we start talking about during conditioning. And the conditioning process is simply us associating that neutral stimulus at the beginning with our unconditioned stimulus and presenting them together, and the presentation of both together results in our unconditioned response. So our tone is presented at the same time as our food, and the dog starts to salivate. Now, at the very beginning, the salivation is pretty much just going to be in response to that unconditioned stimulus. Up front, they don't know anything about the neutral stimulus. It starts off neutral where it has no meaning. And so the salivation at the beginning is an unconditioned response to that unconditioned stimulus. So that's at the bottom here. You have your tone and food, and the dog drools. And you're going to do this multiple times, multiple trials, multiple pairings. So you have your tone and your food presented together, and the dog salivates. Over time, that neutral stimulus becomes a conditioned response. And after conditioning has happened, we can say that that conditioned stimulus is going to produce a conditioned response. I have to make sure I say all of the right words in the right order here. And so our conditioned response is a response that comes to be elicited from our conditioned stimulus. So this is the response that happens whenever they hear what used to be a neutral stimulus, the thing that they have learned to associate with the unconditioned stimulus. So here, after conditioning has happened, you can play that same tone, and even without food being presented, the dog is going to salivate. So in this case, the tone is our conditioned stimulus, and the salivation here is a conditioned response. And so one more walkthrough of all of those stages. Again, this is probably review to you, so you might skip this part, but so be it. Multiple repetitions means it's easier to recall in the future. So before conditioning, we're going to have our unconditioned stimulus elicits an unconditioned response, but the neutral stimulus does nothing. And here we see again that weird notation where they do UCS instead of US. I don't know why they do it. I don't love it. That's why we don't use it. During conditioning, we have this association or pairing between our neutral stimulus and our unconditioned stimulus, and that pairing is still going to result in that unconditioned response because the unconditioned stimulus is still being presented. After conditioning, we can remove our unconditioned stimulus and just present what is now our conditioned stimulus. So our formerly neutral stimulus is now a conditioned stimulus because it elicits a conditioned response, even in the absence of our unconditioned stimulus. And the important part to learn here, to remember, is that our conditioned response is the same response as our unconditioned response. It's still the dog salivating. We change the name of whether it's an unconditioned response or a conditioned response based on what is causing that response. So if salivation is caused by a conditioned stimulus, then it's a conditioned response. But if it's happening because of an unconditioned stimulus, then it's an unconditioned response. So we care about what's causing it to happen. All right, so now that we're refreshed on what classical conditioning looks like, we've talked about the terminology, now we want to talk about researching conditioning. So there tends to be two broad categories of conditioning research. The first of these are people that study the cause or the mechanisms that are underlying that conditioning. Most laboratory studies fall under that category. Pavlov's work fell under that category. But not all research looks at mechanisms. It doesn't necessarily care about the mechanism. Some research is going to look directly at studying functional outcomes or the implications of classical conditioning. And so for the next bunch of slides, we're actually going to talk about two studies, two actual experiments that have been conducted that tried to look at functional outcomes. And so our functional examples are these two studies here. The first, looking at reproductive success. This was conducted by Hollis and colleagues back in '97. And then we're going to look at a study looking at growth rate in grasshoppers, almost forgot the word for grasshoppers, by Dukas and Bernies in 2000. And both of these studies are going to allow us to ask if there's an adaptive or functional value to conditioning. Do they get something out of it? Is there some kind of benefit to it? So let's start with the first of our functional examples, that reproductive success study. Now, Hollis's group were investigating male aggressive behavior as it related to mating success in a species of fish called blue goramus. Again, pronunciation's not fantastic here today, but these fish tend to exhibit aggressive territoriality. Specifically, the males tend to aggressively protect their territories. So they will initially act aggressive towards females until they realize that they're not actually another male. So there's this initial aggression, and then they register, oh, it's a female. Okay, I shouldn't chase them off. I should engage them as a female. This behavior isn't necessarily adaptive because sometimes that initial aggressive stance can cause the female to flee, which leads to the males potentially losing out on mating opportunities. A previous study from other labs had showed that males can learn to predict females through classical conditioning. So we know that they can be classically conditioned, and they can learn that a particular stimulus means that there is a female coming. So as a functional application, this study was looking at testing if males could increase their reproductive success through that classical conditioning mechanism. So we're looking at the function of classical conditioning and how it can change their reproductive success. So at the bottom here, I just have a picture of the fish, and I've left that picture on all the slides to do with this specific study because it helps me separate the two studies in my mind. I also included a picture of Karen Hollis because unlike people like Pavlov, who you have seen a million pictures of a million times over, we don't tend to see some of these other individuals, and it's especially rare, especially in this kind of research, to see women who have done this kind of research. So I included Karen Hollis because she is an awesome person. So let's walk through the methods of this experiment with a brief intermission where I changed a typo, not so much a typo as a lack of a symbol registering. So the box with a question mark in it is just supposed to be an arrow. So I fixed that for us here, but your slides are going to still show the box with a question mark. So as with most of our classical conditioning experiments, everything starts with training. And so this training happened with one trial per day. where we have two separate groups. We have a learning group, and so this PAV just means that there's Pavlovian conditioning in this learning group, and then we have a control group where this is unpaired control. That's the UNP. And so what's happening is in our learning group, they learn an association between a previously neutral stimulus, a light that would be presented in their tanks, that's going to become our conditioned stimulus, followed by a 5-minute exposure to a female, and a female is the unconditioned stimulus. So exposure to a female should cause an unconditioned response, a behavioral response to the presence of a female. For our control group, the unpaired control group specifically, they had 10 seconds of presentation of a light, but it wasn't immediately followed by exposure to the female. In this case, the presentation of the light was independent of exposure to the female. So there's no association here because the stimuli weren't paired. So this isn't a conditioned stimulus and this isn't an unconditioned stimulus for the control group because there is no conditioning process where an association is formed. So randomly they got a presentation of light and randomly they were exposed to a female, but those two events weren't linked, which is different from our learning group. So after that training had occurred, both groups were then given a test where they would have this 10-second presentation of our light, which by this point, because the training had happened, because the conditioning had happened, it should be a conditioned stimulus. And after that, they have exposure to the female. I guess it should be a conditioned stimulus specifically for our Pavlovian conditioned group. It should still be nothing to our control group if everything goes the way we want it to go. So this test is, okay, there's a presentation of the light, and then they immediately get exposure to the female. And the male and female would then be allowed to interact with one another for 6 days, and we recorded their behavior. So what did they do after they were introduced? For our results here, we can look at two different kinds of behaviors that are a measure of indirect fitness. At the top here, we have biting behavior, which would be an aggressive response. And for both of these graphs, we're looking at mean total number of responses, basically how much are they doing that behavior, where higher means they're doing that behavior more and lower means less. And then we have our two different groups here. So we see that our learning group showed a lot less aggressive behavior than the control group. So our control group did a lot more biting, and the learning group didn't show a whole lot of biting. So the control group males acted much more aggressively towards females compared to our learning group, which goes with what we would have anticipated if they had learned to associate that signal with the arrival of a female. And then if we look down here at nest building behavior, so a response that would be appropriate to a female but not to another male, our learning group males performed more nest building behavior. They did this behavior a lot more than our control males. So this is basically telling us that our learning group males reacted accordingly. They behaved in the way that they had figured out that a female was coming. And by having less aggression and more behaviors associated with mating, this is definitely evidence for indirect fitness because those males are doing actions that are conducive to them getting a mate, as opposed to our control group, which did a lot of biting and not a lot of nest building, which is a quick way to scare off a potential mate and a lot less adaptive. Now we can also measure direct fitness benefits, which is basically how many offspring did they end up having. And so we have more graphs here. This is looking at time to spawn, basically how long did it take them to engage in mating behavior. So less time means that our learning group males were a lot more quick to engage in mating behavior. And the control group males took a lot longer to do that. And this is a significant difference here. If we look at behaviors like clasps, so again, mating behaviors, there were more mating behaviors in the learning group than in the control group. And even if we look at directly the number of offspring that they had from this encounter, the experimental group ended up having many, many more offspring than our control group did. So we have evidence of direct benefits to fitness from this classically conditioned association. Now, this study was super important because it was the first real evidence of reproductive benefits that came directly from classical conditioning, which is really, really cool. For our next experiment, another really neat one, we can look at grasshoppers, and as I kind of hinted at already, how they changed their growth rate in based on what they had learned through classical conditioning. So this experiment is going to be looking at fitness-related benefits to learning, specifically in these grasshoppers. And so for our setup here, we have two different types of food dishes. And we could have a balanced diet, basically food pellets that would give them all of the nutrients that they needed to grow in a balanced way. And then we'd have deficient diets, so diets that were lacking some nutrients, so they would eat it, but it wouldn't give them all of the things that they need to grow strong and big. And then we could have each dish paired or associated with a particular color or location or odor that they could then potentially learn about. And so once again, we start off with training, where we have two different groups. You're going to see some patterns here, kind of similar. So we can have a learning group where they were given the opportunity to learn from a bunch of different cues. They could have location, color, and odor associated with either a balanced nutritional supplement or the deficient nutrition. Those would all be constant. So if we look at the bottom here in our learning group, which is the left side here, we have the grasshopper, and in their cage on the left, they always have the color green and the smell of citral, which is a lemon scent, and it would be always on the left, and that would be their deficient food. And then on the right, they would have brown, they would have coumarin, which is basically a vanilla smell, and it's always on the right, and that would be associated with their balanced diet. And so twice a day they'd get the chance to eat, and they'd only be allowed to eat from one of these. But in the morning and in the evening, the learning group had the exact same setup, and it was always predictably presented to them. So because none of these changed, they were able to learn that the deficient is always on the left, or always associated with green, or always smelling like lemon. And they would run enough grasshoppers so that sometimes one group would have it associated with green, some would have it associated with brown, some would have it lemon and vanilla and all of that. So we would counterbalance all of the possible combinations, but for ease of explaining the situation, we're sticking with everything stayed in the same place, just like this. For our random group, in this situation, everything was randomized. So the location would randomize. Sometimes the deficient was on the left, sometimes it's on the right. The colors could be randomized. The odors could all be randomized. So we can control for each of these individually, where maybe you'd hold some of them constant and not others. But basically this group, because of that random changing, they prevented those grasshoppers from learning any kind of association. So they couldn't predict which of the plates would have the deficient food and which would have the balanced food. At least that's the theory here. So let's look at the results. These graphs at the bottom here have proportion of visits to balanced diet, at least for this first one here. And this is going to be in time. They're looking at age of the grasshoppers, but whatever, time is time. And it's important to note here that 0.5 would be about chance levels. So if you have a 50-50 chance of choosing between two options, you're sitting at chance levels. And so we can see for the green line, our learning line, it took them a little while to figure it out. At first, the green and the orange are pretty much the same, but after about 3 or 4 days, they've figured out the association and know which side or which color or which smell to go to. And these are the proportion of visits to the balanced diet. So 100% of the time, pretty much, they were visiting the balanced diet plate. They went to the more desirable of the food types. Whereas our random group, this orange line here, hovered right around chance levels. They both started around 0.6, but that isn't necessarily far enough away from chance levels that we should be concerned. And then everything kind of sits here. It's hard to tell what's a significant deviation without error bars and stuff, but we'll just keep that in mind. Now, the other graph showed us something interesting. So again, we have time on our x-axis, that's not different, but on our y-axis, now we have proportion of time feeding on that balanced diet. So not just number of visits, but the proportion of time. And so this basically shows us that both ended up preferring to spend more time on the balanced diet. So they liked to eat the food that was more beneficial to them. So even the random group, when they found that balanced diet plate, they spent more time there eating that food, but because they didn't know which side to go to or which color to go to or which smell to go to, it took them longer to figure out which plate they should be going to get the best food. Now, in the wild, this ability to learn and having this kind of association learn has some logical adaptive value. Because if you know which food to go to or where to go to it or some other feature that will lead you directly to the best food source, it means that in the wild you would spend less time moving, you'd waste less energy moving around and searching different areas. Because you're moving less, you might be less visible to predators. There are all sorts of potential benefits to having that kind of knowledge. So from an adaptive point of view, it makes sense that learning would be beneficial. And if we want to look at the actual fitness of these organisms, we can look at our two treatments, our learning group and our random or control group, and the growth rate. So the idea here is that a higher growth rate means that they're probably eating better food and they're healthier, therefore more fit. And our learning group, it had a much, much higher growth rate than our random or control group. In fact, our learning group ended up growing about 20% faster than the random group, which is a massive increase in fitness. And this study and others that sort of stemmed from it showed lots and lots of different variants of this. They also found that growth rate is positively correlated with egg size and number of eggs laid. So an even more direct connection to fitness there as well. So having that knowledge and being able to use that knowledge is beneficial in a bunch of different ways. So another very, very cool experiment, especially because we're looking at such a small, short-lived insect, which, at least when I started out studying learning, I hadn't expected much of what we would have called quote-unquote simpler organisms. But the more we learn about learning, the more we realize that it isn't something that we just see in the large, fancy mammals. We can also see it in insects and other animals that we might not have considered to be capable of it, which I love sort of turning over some of our assumptions and preconceptions. All right, so we're going to go back to some of the more textbook specific topics, and we can talk about some of the classic experimental paradigms, which are basically the four main ways that we've seen people study classical conditioning up to this point. Based on the way that I've divided up my slides, we're not going to get through all of these. We're just going to get started and talk about fear conditioning, and then we'll talk about the rest of them in the next class, just so you know what we're expecting here. So let's talk about fear conditioning. This whole idea is based around the thought that fears are learned and can be conditioned just like any other kind of behavior. And this started back with Watson and Rayner back in the 1920s. Again, because you've come this far in psychology, you have absolutely heard about these two, and the little Albert study. So I won't go into too many details because you've already done this before. I've also posted a video going over the little Albert study just in case you want a few more details, but I won't bore you with this. Basically, they took a 9-month-old infant named Albert and trained him or conditioned him to experience fear. So he had fear conditioning, and it was specifically to a white rat. And this white rat itself was docile, but they conditioned that fear response by pairing the presentation of a white rat, which started out as a neutral stimulus that the baby didn't mind, but by associating it with things like a very loud noise that startled the child and made him cry, they could then make him cry just whenever he saw the white rat. This is also a great study to talk about things like generalization, where he came to associate anything white and fuzzy as being scary, but that's something for another day. But this study, as controversial as it has been, was the first evidence that we had for classical conditioning of emotional responses, fear being one of the most potent emotions that we can experience. Fear and anxiety are sources of intense discomfort for humans and probably for other animals as well. If the experience of those two emotions is severe enough, they can lead to serious psychological and behavioral problems. So we can talk about phobias, where people can have intense fears tied to particular stimuli. And when these phobias develop, we can usually see some kind of initial trauma where an unconditioned stimulus and unconditioned response sort of come to be associated with what otherwise would have been a neutral stimulus. So standard classical conditioning setup, but with something that shouldn't necessarily be linked with that unconditioned stimulus. So we can use the example of somebody developing a fear of dogs. So their initial trauma is maybe they were bit by a dog when they were younger. The unconditioned stimulus being bit by a dog, the unconditioned response being pain and fear. The new stimulus, the conditioned stimulus, would then be the sight of dogs. And if they've generalized, it could be any dog, even very small, cute dogs. But that association between that conditioned stimulus, that now conditioned stimulus, and that unconditioned stimulus from that initial trauma leads to a conditioned response of intense fear. And this can be considered irrational because that conditioned stimulus used to be a neutral stimulus that might not actually be directly related to that initial trauma itself. So it's kind of hard to pick these apart because they just sort of happen. Now, the detrimental nature of phobias and other kinds of fear conditioning in humans, it definitely makes it something that people are interested in studying. So how can we actually study this? In non-human animal research, they tend to focus on sort of how is that fear or anxiety acquired, sometimes specifically looking at the neural mechanisms that underlie that acquisition. Now you might be thinking to yourself, how do you measure something abstract like fear in a non-human animal? If you're looking at it in humans, you can just ask them, on a scale of 1 to 10, how afraid are you? But you can't ask a rat how afraid they are. So instead, we have to measure fear in an indirect fashion in other species. A really common example of this would be freezing behavior in rats. So freezing is an adaptive defense response that has evolved in rats so that if there's something that startles them, if there's something that is painful or uncomfortable, they'll freeze, they'll stop moving, and they'll hold very, very still. So we can actually measure their rate of movement before and after a particular stimulus to see what degree of fear are we noting? So freezing behavior is an indirect measure of fear. Now, the standard setup for fear conditioning is we have a conditioned stimulus, something that starts as a neutral stimulus, as per always, something like a tone that we then pair with an aversive, unconditioned stimulus, something like a shock or a very loud noise or a very bright light, something that is uncomfortable. Now, I always like to note to people that when we talk about things like shocks, especially in terms of, say, fear conditioning, these shocks aren't meant to be something that is massively harmful or even overly painful. They're just meant to be uncomfortable. In a lot of laboratory setups, when they use shocks as an aversive stimulus, they're usually done in a very controlled way, and they're only as intense as is required to cause a bit of discomfort. They really want to avoid physical harm. When it comes to ethics of animal research, one of the main tenets is to reduce that harm, to try and do the littlest damage as possible. So even if studying fear is something that has been deemed as really important, and therefore you have ethical approval to do it, you want to do it in the least harmful way possible. So a lot of researchers will use something like a loud sound or a bright light, something that doesn't cause a sort of physical stimulus, but it sort of depends on the situation. But I just know that sometimes when people see the term of applying shock, they think of some massively painful shock, and that's absolutely not what happens in the real world, or I guess in the research world. It's usually just something that's enough to startle them and to trigger that freezing behavior and nothing more. All right, so if we're actually going to measure this freezing behavior, how do we calculate that? We can use something that's called a suppression ratio. And a suppression ratio is basically going to take the amount of behavior or responding that happens when the conditioned stimulus has been delivered. So if the shock or loud sound has happened, how much activity are they doing? And you're going to divide it by a combination of that behavior that they elicit or that they do after the conditioned stimulus has been presented, plus how active they were before that conditioned stimulus was presented. So the pre-conditioned stimulus behavior. So if we're looking at rats for movement behavior, we can get a measure of, say, a distance per minute when the shock or loud sound has happened, divided by that amount of movement, sort of distance per unit time, after the shock has been delivered, plus how much they were moving before the shock had been delivered. So by including that pre-conditioned stimulus delivery behavior, we can see how it's changed from before the thing of interest, our conditioned stimulus, has happened. And it doesn't have to be freezing behavior. You could look at something like lever pressing. If you have rats in a box, you can look at how often they press a lever after a conditioned stimulus has been delivered versus before the conditioned stimulus has been delivered. And you would see sort of reduced responding to that lever pressing if they're showing some kind of fear response. And so these values, if we calculate out a suppression ratio, we can go anywhere from 0 to 0.5, 0 meaning that there is complete suppression, and 0.5 meaning that there's no suppression at all. And smaller values closer to 0 means more fear because there is more behavior being suppressed. There's more of a disruption of their natural behavior. Now, for those of you who don't necessarily understand concepts well from just talking about them, I also have an example with numbers in case that helps. So here are 3 different examples. We have everywhere from complete suppression to partial suppression to no suppression at all. So let's start with the middle ground here. And if we want to look at our responding, let's use that example of pressing a lever. So for our less pressing during the conditioned stimulus than before the conditioned stimulus was presented, maybe when that conditioned stimulus, when that shock has been delivered, we only see 5 lever presses per minute. But before the shock was delivered, they were pressing the lever 10 times per minute. So we take that 5 times per minute and divide it by 5 plus 10 times per minute, and it gives us a 0.33 suppression ratio. So that's somewhere in the middle. It's between 0 and 0.5. So we're saying that there was some suppression, but it wasn't complete. So it's somewhere in the middle. For complete suppression, we would be looking at something like, say, zero lever presses. Our pre-conditioned stimulus responding is still 10, so that number stays at 10, but now our after conditioned stimulus delivery behavior is 0 lever presses. So 0 divided by 10 is still 0. That tells us that there was complete suppression. There is no behavior after the conditioned stimulus was delivered, even though there was some before it was delivered. And if we see no change in pressing, we would see that before we still have 10 lever presses per time, but after that conditioned stimulus was delivered, we would see 10 lever presses, which is the same. So 10 divided by our 10 plus 10 is 0.5. So that is the largest of our suppression ratios that we can get, and that tells us that there was no change in lever pressing during the delivery of that conditioned stimulus, so there was no suppression of behavior in that example. So hopefully that helps you kind of visualize what's happening and how the behaviors are changing. All right, and then the last point under fear conditioning is to make mention of conditioned emotional responses in general. Emotional responses are more than just fear. Fear just happens to be a really easy one to talk about because fear tends to be a fairly biologically relevant emotion, so it's fairly easy to study. But we can look at conditioned emotional responses to all sorts of emotions, not just fear. And we can see lots of conditioned emotional responses in our everyday lives, especially through advertising. Advertising uses the association of emotions, things like happiness or contentment, with previously unfamiliar stimuli, things like brands, by way of a familiar, already existing conditioned stimulus. And in this case, this existing stimulus would be an emotional stimuli, something like parties. So you associate parties with the feelings that you have when you go to a party. So alcoholic companies would, companies that sell alcohol is probably the more accurate way to say that in English, want you to think of exciting and happy and good things when you see their alcohol, so they would advertise with pictures of attractive people at parties enjoying themselves, so that you then associate their product with also enjoying yourself. This might remind you of that idea of second-order conditioning, which is basically what this is describing, which I'm sure we will talk about quite a bit more as time goes on in the course. But some great examples down here of branding that's trying to associate their product with things that make you happy. So suns and pretty colors and happiness even right on their advertisement. Or Budweiser and look at these cute animals and their friends, and that makes you feel warm and cuddly, and now you associate us with warm, cuddly feelings. So that's conditioned emotional responses. So we're going to talk about the next 3 of our, there we go, classical experimental paradigms, eye blink conditioning, sign tracking and goal tracking, and taste aversion learning in the next class, so for Thursday's lectures. We'll see you guys there.